Okay, so to continue on with, and actually we'll finish chapter 9 here. This will also lead into today's lab, but just to finish this little note here, when we talk about simple dominant recessive inheritance, we can use a dominant allele A and then the recessive allele, the little a. We'll be using those in quite a few labs. But when we talk about the recessive phenotype, that means in order for them to show that recessive characteristic or that disorder, they have to be little a, little a. Now, the dominant individual, the dominant phenotype can either be homozygous dominant, big A, or heterozygous, big A, little a. We tend to refer to some things as wild type traits, and that doesn't necessarily mean it's the dominant trait. It just means it is the most prevalent, the one we see most often in nature. And then here's a few human traits when we look at them, like freckles, believe it or not, it's dominant over no freckles. And then the widow peak, that's where the bangs kind of go down into a point on the forehead, like Dracula. That's dominant over a straight hairline. And then we have earlobes, whether they're free or they're attached. So those are just some of the characteristics there we see in humans. So when we talk about inheritance patterns, we're going to talk about whether it's a recessive or a dominant inheritance pattern. If it's recessive, that means you have to have two recessive alleles, two little a's, to show the disease. So you can have two heterozygous parents that look perfectly normal, don't have the disorder, and because they're both carriers, they're both heterozygous, they could have a child that has that disorder. And so this tends to increase in frequency with inbreeding. When you have groups of animals, organisms, I don't want to just say people, it could be dogs, cats, horses, whatever. When your population size is small, it increases the number of potential carriers in the population, which then would increase the frequency or the occurrence of there being individuals that have that recessive disorder. Now, dominant inheritance means you only need to get one bad copy and you have the disease. And so, dominant alleles that are lethal tend to be eliminated from the population. Not that all dominant alleles are lethal, but ones that are, you get one copy and you die, therefore you can't pass it on. So there are a couple different things we're going to talk about with dominant disorders, a couple different dominant disorders. So here we just have, let me make sure I didn't skip one. Yep. So here we have two heterozygous. Now they're normal. So this, of course, would be an example of our autosomal recessive. So when we look at this, we notice all three of these individuals here, okay, are normal. Now we have one that is homozygous dominant, which means that they're not a carrier. They will not be giving a bad gene to their children. We have 50% that are carriers. They're normal. They don't show the disorder or the trait we're looking at, but they do have the potential of passing it on to their children. And then we have our poor deaf individual here, little d, are homozygous recessive. So again, this would be an example of our autosomal recessive. When I talk about autosomal, I am talking about it is on chromosome 1 through 22. Everybody has two chromosomes there, right? Two chromosome 1, two chromosome 2, one from mom, one from dad. You know, we have 23 pairs of chromosomes, and when we get to that 23rd pair, that's those sex chromosomes. And we're going to talk about those a little differently when we get to that segment. But right now, I just want to focus on the autosomals. That's the genes on chromosomes 1 through 22. So one of these examples that you may have heard of is called cystic fibrosis. So cystic fibrosis is a disease that there is a salt transporter. It's a protein in the membrane of the cell membrane, and it allows for salt balance so homeostasis with our salt concentrations. And when it's not made, that transporter is not made correctly, you failed to maintain homeostasis. And because of that, remember we talk about water follows the salt. Well, that's what happens. And then we start to secrete this thick mucus. And they tend to get this in their lungs and in their digestive organs, specifically their pancreas. So it is recessive. It is carried by approximately 1 in 31 Americans. And so when we see people that have cystic fibrosis, that means both parents had to be carriers. They're both carrying that disorder. It used to be that you were lucky to live into your teens with cystic fibrosis, but with today's medical interventions, they're living into their mid to upper 30s. So this has gone a long way with care. Now, the dominant disorders that we'll tend to talk about, there's 2 of them here. One is called an achondroplastic dwarf. Now, with achondroplasia dwarfism, that is the most common dwarfism. There's like 5 different types of dwarfisms, but this is the one that you're most familiar with. So this is the kind of dwarf, like if you watch Game of Thrones, this is what the dwarf on that show had. So with achondroplastic dwarfism, it is a dominant disorder, which is why, and it's kind of interesting because homozygous dominant, so if you had big A, those homozygous dominant in achondroplastic dwarfism is lethal. So they are generally miscarried or don't live long after birth. So all the achon dwarfs you see are heterozygous. That means they have big A, little a. That means they have a potential of having a normal child. And so when you could have 2 achondroplastic dwarfs get married, have kids, and have normal sized children because they both are carrying basically that gene for normal bone development. And then we have a disease called Huntington's disease. Now, Huntington's is a disorder that basically starts to deteriorate the brain. With Huntington's disease, again, and you only need one bad copy and you have this. So with Huntington's disease, what happens is you get into your 30s and you start showing symptoms. So the problem with this particular disorder is it used to be that you would end up passing on to your children because you didn't know you had it. So with today, we can do what's called a pedigree, and we can figure out what is the potential that you have one of these bad copies. And now with genetic testing, because we've identified the gene, you can be tested and they can determine whether you have it. So that might be a decision that you want to make if you want to have children. Because again, usually you start showing symptoms in your mid 30s, and you usually only live about 10 to 15 years once symptoms start. So it would be kind of sad to have kids and then die early in your lifespan, or pass that gene on to your children would be even more problematic. So here is a chart of a bunch of different types of disorders. So we have albinism, and that these ones are the recessive ones. So you need 2 bad copies to be an albino, and again, you lack the pigment melanin, so you don't have any skin. Your skin's really white, your hair is white, your eyes are red because you don't make that melanin to make brown or blue eyes. Cystic fibrosis I talked about. Galactosemia is one where galactosemia is a sugar that you're missing an enzyme to break down galactose, which is a disaccharide. And so without this, it actually accumulates and causes some tissue damage. They've used that mental retardation. I know we keep going back and forth in our politically correctness, what word to use, whether you want to use cognitive disability, but their brain function is not normal. And then eye and liver damage. And that one, again, if you eliminate galactose, so this one's very similar to PKU. So with PKU, it's not a sugar, it's an amino acid they cannot break down. And so this also accumulates into the bloodstream. So with both galactosemia and PKU, if you avoid those particular things in your diet, you will develop normally. So we need to be very careful with those. And then sickle cell disease. Now, sickle cell, we've already talked about a little bit. And we'll get into some of that stuff when we talk about evolution, how it is actually a positive mutation if you're living in areas with malaria. So we have sickle cell disease and then Tay-Sachs. Tay-Sachs is another one where what happens is lipid accumulates around the nerves in the brain, and it basically suffocates the nerves. And so these babies, it's really sad. They start developing very normally, and you would know they roll over, they sit up, they start cooing and doing all the things they're supposed to be doing. And then all of a sudden their development starts to regress, and they go backwards, and they go to the point where they can't even breathe, and then they die. So most individual with Tay-Sachs, I think that, I don't want to say the average, but they can live up to one. I think the average, I mean two. I think the average lifespan is about one. So really sad. It's really high in the Jewish, especially the Ashkenazi Jews. They tend to stay within the religious group, marry within that group, and so their population size is small, and they've got this gene fixed in that population, so they have a large number of carriers in that group of people. So it's really kind of sad. Dominant disorders, I already talked about achondroplastic dwarfs. And then Alzheimer's, there's different types of Alzheimer's. So, but there is one type of Alzheimer's that's a dominant. I talked about Huntington's. Hypercholesterolemia is when you have too much cholesterol in your blood. Now, there are many different reasons for having high cholesterol in your blood. So this is just one particular reason. And this is where you might have a person who is very fit and lean. They eat, really, really well, low fat diet, exercise, do everything they're supposed to do, and they still have high cholesterol. And it could be because of this genetic reason, and which of course then we have to get on medication to control that. So this is just kind of a rundown of some of these human genetic disorders we see. All right. So let's go into pedigrees, and that's kind of what I want you to focus on. Now, I want you to focus on the autosomal pedigrees at this point. We will get into our lesson on sex linked genetics, at which point then I will introduce the sex linked pedigree. So let's just focus on the autosomals at first. So a pedigree is where we can take kind of a snapshot of a family, and we can then follow traits along those individuals and kind of see what kind of inheritance pattern it is and what's the likelihood of someone having it. So again, I'm going to talk about autosomal recessive, autosomal dominant, and we'll do sex linked later. So here is an example of an autosomal recessive. Like I said, already talks about, it's on chromosomes 1 through 22. You have to have 2 bad copies. Now I'm going to try to write here. I was hoping this is kind of filled in a little bit for you. Now I know I couldn't find a ton already made with S's, but it doesn't matter what letter you use. Okay. So, but what I'm saying here is with this particular autosomal recessive. So remember, with autosomal recessive, oh, it's not cooperating. All right, so if we have a heterozygous, 2 heterozygous, the only way you could have the disorder is for little b. So notice right here, the squares, now the squares are males. The circles are females. And so, and if it's shaded in, that means they have the particular trait or disorder we're looking for. That's what we're studying. So for instance, let's just say this is for sickle cell anemia. All right. So this girl here, she has sickle cell anemia. Now over here, we use the Roman numerals for generation. So this is generation 1, this is generation 2. And then if they have a child, we'd have 3 generations. And then we can also, I think in your lab book, there's 2 different ways to number individuals. And that is you can individually number them, one with the Arabic numbers 1, 2, 3, 4, 1, 2, 3, 4, and then you go 1. So you'd start with 1 at every generation. That is one method. The other method, which is what we use in our lab book, is just numbering them start from finish. So 1, 2, 3, 4, continue over here, again, going from left to right, 5, 6, 7, 8, and then this would be 9. right? So a couple different ways to do that. And if you go on internet, like I said, you'll see both. So if I ask you to fill in, so the phenotypes, let's just use some vocab here. The phenotype is what we see. So right now, this person's normal, this female's normal, this male's normal, this female's normal. This female is not normal, has sickle cell. This one does not. This male has sickle cell. Now, if I ask you for the genotype, that means I have to put the letters in. So I was hoping I could do S's here. So with little s, we know they get each one of these letters from one parent. So we know this girl here, and the girl is older than the boys because we start with the oldest on the left. So the girl, they had a daughter first, and she has sickle cell anemia. That means the parents need to be a little s and a little s, right? So each of them gave one of their little s's to their daughter. Now, he does not have sickle cell anemia. This is where it gets a little tricky because we know he's got a big S. Now, I don't know if he got the big S from mom or dad. I just know one of them gave him a big S. Now, what is the potential of giving him what could be the second gene? Now, remember, he's normal, which means he could be big S, or big S, little s. I don't know what he is. So I generally put a question mark when I don't know. Don't leave it blank, because if you leave it blank, I just think you just didn't fill it out. Now, you could write this as big S, or big S, little s, because it could be either one. Or if you put the question mark there, you're telling me, well, it could be a big S, it could be a little s. I don't know. Not enough information to tell that. All right, so maybe if they had a child here and he had sickle cell anemia, then I would know this individual's big S, little s, but I don't have enough information to finish that. Now over here, I know the parents again. So the parents are normal, so I know they have one big S. But they also have to have a little s, right? Because they gave their youngest child, which is a boy, sickle cell anemia. So we got a little s. And again, I have the same exact situation over here. I don't know what this one's going to be. So I got to put a question mark. Okay. All right. So that is autosomal recessive. So normally with autosomal recessive, what I look at is if I have two normal parents have a child with it, it's got to be autosomal recessive. Okay. That means they're both carriers. They're hiding those bad genes. Versus autosomal dominant, which is what we're going to look at next. So with autosomal dominant, again, it's on chromosomes 1 through 22. In this case, if we look at it this way, all three of these individuals right here, they have the disorder. They have it. Okay. So let me think, let's see, what genetic disorder? So I'm going to use, I would do baldness, but baldness is tricky because it's sex influenced, which we'll talk a little bit about that later. Let's just use achondroplastic dwarfism. So we're going to say this is dwarfism. So all three of these are dwarfism, which again, this one's a little trickier because I couldn't, big B would die, but we're just going to assume they're not dead. All right. Just for lack of confusing this. Okay. Or let's just do this. I'll switch it to cholesterolemia, even though it's a hard word to say. So high cholesterol. They are healthy. They don't have high cholesterol. So they're normal, right? All right, so notice I have two parents here, okay? Now they have high cholesterol. Now, how can they have a normal child if let's just say if this was autosomal recessive, that means this would be little b, little b, little b. There is no way they could have a normal child. So I always look at, like, if you have two people that have the disorder and they have a child that doesn't, it's got to be autosomal dominant. Because what I'm saying here is this individual is little b, which means this parent has to have a little b. This parent has to have a little b. And we know both of them have to have a big B because they have the disorder. So I always feel like the dominant ones are easier to fill in because I know everyone that's filled in, they have the disorder, so they have the big B. This is so hard to do. So they have the big B. All right. Now, all the ones that are blank don't have it. The only genotype they can have is little b, little b. So all of these are little b, little b. Okay. Little b, little b. Okay, so the ones I have to fill in now are these ones over here. So this one could be big B, and this one could be big B, but they also could be big B, little b. I don't know. I'd have to see their children. So this is another instance where we're going to use the question mark. Or you could write big B, or big B, little b. Now, what about this over here? Now, this one over here, they have a normal child. So, right, this parent gave one of the little b's, right? So the other one has to come from this parent. So this parent has to be heterozygous. Now, this child over here also has to be heterozygous because you got the big B from dad, right? So, but mom only has little b's to give. So she gave her son a little b, right? So that would be how you would fill in an autosomal dominant pedigree. I always like them because they're like little puzzles to fill out. Okay. So sex length, like I said, we'll do that one later. And then the last thing this chapter talks about is how we can possibly detect some of these before you have a baby, during pregnancy, and after birth. So we can do a lot of genetic testing to identify these parents. So as long as we've identified these genes, we can find out whether you're a carrier, whether you have it, you know, what's going on. Like I said, if Huntington's, you got to figure out, do you have the gene? You may not want to have children. All right. So what we can also do here, or you could maybe find out if you have the gene for cystic fibrosis, and if your husband does or your wife does, and you both do, you might decide, there's a 25% chance that you would have a child with cystic fibrosis. So these are all decisions you may need to make. Now, once you are pregnant, so during pregnancy, and hopefully you don't have to deal with this. I know we do a lot of tests. I know many of you already have children, and you know that. I always say that every time you go in for a test when you're pregnant, they're drawing blood. And they're always saying, well, we want to see if you're anemic. And I'm like, well, if I wasn't before, I am now, because you're constantly taking blood from me. And it seems like they still do that, because I hear from pregnant people today that say, oh yeah, they drew blood again. So they're doing all these, and now they can do all these extra tests with blood. So it's amazing what they can do with blood tests. So what they'll do with those blood tests is generally they'll get some basic information, and then they'll determine whether they need to get some cells from the baby, because once they get cells from the baby, they can actually then determine if they have one of those bad genes, if they have cystic fibrosis, if they have Huntington's, all these different things. So there's two types of tests they can do. One is called an amniocentesis. And what they do is they take out the fluids. They put a needle in through the belly. And actually I have a picture here. So I'm just going to go straight to the picture here. So what they're going to do is they're going to, here's the amniocentesis. So they put this needle through the belly of the mom into the uterus, and they draw out that amniotic fluid. Inside that amniotic fluid are cells from the baby, because we're constantly sloughing off skin cells. And so once they do that, they can then take those cells and culture them, and then they can do a karyotype. We've already studied what karyotypes are. And during a karyotype, they can tell if there's any gross chromosomal abnormalities, maybe Down syndrome. They do that a lot with the Down syndrome. If your alpha fetal protein test comes up funky, your ultrasounds don't come out quite right, they may want to do an amniocentesis. They can also label and do some tracking for specific genes on these karyotypes as well. The other type of test they can do is called CVS, chorionic villus sampling. And with that, they're going to go through the birth canal, and they're going to take basically cells from the the chorionic villi, which is kind of a part of the placenta. The placenta and the villi, the chorionic villi, they're all made up from the same cells that make up the baby. They're baby cells. So, but the difference between those cells and the amniotic fetal cells is that the amniotic fetal cells tend to be dead cells that we have to culture, and the fetal cells are much more viable and healthier. We get those things replicating and dividing very fast. And so with the CVS test, you can get your results back. Notice it says several hours versus weeks. Okay, so we can get those results back much faster, so that if there's decisions that you may want to make, you have that option. So a couple different tests. Now, they will tell you with both tests, there is a risk of miscarriage. So there's a lot of decisions that have to be made. And hopefully none of you have been in that position where you've had to make those decisions or have those test runs, because they're very scary. So here's just a little bit more. I already kind of mentioned the blood tests. They do a lot of different blood tests to identify these genetic abnormalities. And then ultrasound does a lot of stuff. We can see that on ultrasound. So we have a lot of different tests that we can do. And so again, with these new technologies, we have ethical consideration. We have confidentiality of this genetic testing, the cost. Again, with your results, what do you do with those test results? You know, there are a bunch of different decisions you may want to make. I know some mothers don't want to have, and you can refuse to have the test done because there is no decision to make for you. Although I know some moms might want to at least get prepared for what they're going to have to deal with if they do have one of these children that have a genetic disorder, so they can be educated and know exactly what they need to do to keep that baby as healthy as possible with whatever genetic disorder they happen to have. So I believe that is the last slide. And next, I guess the best thing would be to move on to your lab, or you could go ahead and start the next genetic presentation.